Yeah? Fantastic. That was just the call you spotted it immediately. We didn't, I didn't miss 15 minutes or something.
No.
Great. Okay. Well, as I go just back in all these lovely slides and tell the whole story again, you would lovely forget that. Here's all the time. Okay. We can coming up. Secondary structure guided alignment. So basically, there are a lot of programs that can use parts of structural features of proteins. Does any one of you remember this sentence in evolution? What is more conserved than what? Structure more conserved than sequence. Now, so if we are able to really predict these things well, or even better to observe structural features, if you can use that, that should help, of course, right? But we are going to use predicted information. How good is the prediction? Okay, so here is a slide on that. So here's your sentence. I checked you beforehand, but you were perfect. Structure more conserved than sequence. So secondary structure prediction at the moment is at the level of 80% correct prediction. If you predict 5 amino acids, the secondary structure of that on average 4 out of 5 are correct. 1 out of 5 is incorrect, right? That's your error rate. And so, does this help or not? That's the question. You know, you would agree with me. If 1 in 5 is correctly predicted, then using that information is a bit iffy. But 4 out of 5 turns out is enough, is good enough. You'll hear a lot about this in the next course, Structural Bioinformatics, or that's starting in February. The next course will be another course in January. So tertiary structure, so predicting 3-dimensional protein structure is still quite a challenge in some cases. But it's becoming also better that we can start using it. I will now talk about using secondary structure prediction. Now, how about this slide then? Is that of any use to do this? And here's a slide that sort of tells something about that. So, this is a while ago, but you know, secondary structure prediction methods is also, you know, people develop methods to do it. There are maybe 20 or 30 methods to do it out. And, you know, people try and argue better than me and so on. And there are even whole competitions called CASP. Every other year, people go to a nice place in the world, mostly in California, where they sit together. They get sequences they need to align or sequences they need to predict, but people have made a real structure, but they're kept hidden. So scientists, crystallographers, and people do and are really solving protein structures, observing protein structures. They get a telephone call. We hear there's a structure coming out. Would you mind not publishing it after the meeting? We write 1 extra paper, and you'll be a co-author. So they wait. They say, okay, then I'll wait tomorrow. So that's kept secret. So then all these guys are running their programs and brag about it. I'm so good, and you know, this is fantastic what I've made, and you'll see in September. And then in this conference they sit together, the real answer comes out, and of course you're terrible and much worse than you thought. And then the idea is, in the early ideas they did something stupid, I thought, because then you would go, why is this guy so good? So you walk to the car. What are you doing? You're so much better than I am, right? And then you learn, you try. That was not allowed initially. You had to use your feet. Everybody, it was a discussion, and how did you do this? That was not allowed. So that didn't really work. So now you can. So they have all kinds of topics that predictions are being done, and they use all of these structures that are hidden, and then after the meeting, all these structures are published, and you know, these authors can use the publication. We joined 1 year, and you know, it's always a good thing. We just looked regardless. There should not be a structure of here, but we looked and we found the author had, without knowing or realizing, put the 3D structures on the web. So you were perfect on that one. So amazing. Anyway, that's also information. You're allowed to use the information that is there. Okay, is that cheating? Anyway, so people are doing this. So over the years or so, you know, initially it was like 80%, now sorry, 60%, or you know, so it crept up now to 80, but 10 years of work was maybe 5%. From 65 to 70 was a hard battle, right? All kinds of new ideas all the time. Now, if what you just could do, we did a simple thing. We said, okay, there are a number of secondary structure prediction methods. You know what? We have made this method to use it. We just use the information from this method, make an alignment, using the information from another method, secondary structure, make the alignment, and now let's compare these alignments. So these programs, the secondary structure prediction programs, had a difference of 5% in quality. the difference in alignment quality was huge, 40%. So what you really use as extra information is really critical. But makes a big difference. That's the good news, right? You have to be careful, but it really can help. So basically, there is something here to do. So we talk about, so you can do all kinds of things, so amino acids, secondary structures, super secondary structure patterns, or all kinds of other things. You know it's a hydrophobic region, now you use some other probabilities for matching, all kinds of tricks you can try. People have tried many things. I'll zoom in on secondary structure now. So how are we going to put in this information? Anybody an idea? Oh, it's Friday, I'll tell you, okay. So we call this secondary structure guided praline. And I'll show you now a picture how we use the information in praline of secondary structure. You know what is the advantage of secondary structure? I show you here. It's just also a string, you know. The input sequence are strings with 20 types of letters, amino acids, and here you see the encoded string for the secondary structure. Next course, guys, so bear with me, I'm not talking all the details, but basically what most prediction programs do, they predict helix or strand or not helix or strand, which is called coil. Now, the coding for H you can guess, that's helix, right? A beta strand is E because a beta strand is the most extended possible structure. Next course, the letter E is for beta strand and C is for coil 3. So here you are. Now, what could you use? How could you use this information? Now, some programs they say, what we do, that sounds clever. We also thought gaps are less likely in secondary structures. So if you have this extra information, you can say, you know what? I'll hire the gap penalties for these amino acids, and I'll hire them for these, and I keep them low for not helix or strand. You differentiate gap penalties. That's one possibility. We do something else. We say, look, we have databases full of sequences. We know which part of protein sequences are in helix. We have many we observe. We know where the secondary structure is. We said, okay, now we have a helix in this sequence and a helix in this sequence. We know the alignment. We know that these two helices are aligned. Now we can talk about what rules of exchange, what amino acids can evolve in a helix. Would that be the same as in a strand? Would it be the same as in a coil region? And the answer is no. The evolutionary rules are different. So people have taken steps, other authors have done it, we've made these matrices ourselves. They've looked in databases, compared all helix-helix alignment, and compiled a blossom matrix, quote unquote, only for helix. Secondary structure specific exchange matrix. This is the default blossom matrix that we explained a few lectures ago. So you do this for each of the three possibilities, for helix, for strand, and for coil, for not helix and strand, have these matrices. So here is one sequence, here is the secondary structure string of that sequence, here is the first three are in helix, then 3 in coil, and the last 6 are in strand. Here it's a bit different, the helix is a bit longer here, 4 long, and 2 coil, and 3 strand, and a bit of coil in the end. If you align it, what do we have here in the matrix? The region where the helix is aligned onto a helix. You see that? H against H here. This is C here, CCC here, CC there, and this is the strand region in the one sequence against the strand region in the other. Now, we use this information, we use the exchange probabilities from these specific matrices, and if there is something, here is a helix against coil in this sequence and against strand, we don't know what to use here, we just use the default matrix as we always do. Normally, right? So, does this help? That's the question. And the answer is yes. Not as much as the psi blast trick, but this also helps. So basically, here is an alignment where you see, you don't need to read all the letters, it's impossible. It's also a faded slide. You see here this string with the secondary structures, and you see what praline in those days, it's a while ago this one, makes of it. And so you see here EEE, it's very small, but maybe some with very sharp eye see it. That's strand, so it's strand, helix, strand, helix. This is the same alignment, by the way, of the plate reductions you've seen a few times before. Even the key Y's, the outlier sequence is here. And here you see, there's a lesson here. Do you see this region here of strand? Do all sequences predict something? You see empty here? No prediction of the strand there. It was not predicted. Here, this helix bit is predicted in all the sequences. Do you see this region of strand? No, no. Shifted. Okay, so this is prediction. Sometimes you are able to predict, sometimes not. But that information is used and the alignment is made. Yeah?
But the level of the region, helix, column,
this one is based upon.
When done.
Before we do the alignment, we predict for each of the sequences, each input sequence, we predict the secondary structure. So what you see here is the result of running the secondary structure prediction program.
Using only the second
So many of secondary prediction programs take a sequence, run side last, accumulate sequences, then look and predict. So they all have their own tricks.
But we need the PDB file or something
No,
about structure?
PDB file will be perfect because they have the three-dimensional structure. I mean, you really know where the helix is, and they don't need to predict if you have that. So this program is clever enough, by the way. Okay, to check, this is a sequence. Does that sequence have a 4-letter code? Yes. If so, would this be a PDB identifier? If so, you take the secondary structure from PDB rather than predict. No need to predict if you know it. No? But many sequences here, and basically if you look, there are 4 sequences. If you want to know here, you see that? I think there are 4, 1, 2, 3, 4 sequences that have a structure. We use that. For the others, we predict the no. Yeah? Okay, so it's, I said it works, it optimizes, but not always, and to a lesser extent than type writing. Okay, using this technology. Who have heard about transmembrane proteins? You know what that is? These are proteins that are not just floating, if you like, in the cell. They are connected or even embedded in the membrane, in the outer wall of a cell, you might say. You know what is the advantage? What do you do if you go out, you want to meet people? What do you do? Oh, you go on WhatsApp nowadays. But in the old days, we would go into a bar and meet people standing at the bar. You know what is great about the bar? If everybody stands there, you have 2 neighbors. You can say hello. You optimize the chance that you meet somebody because a lot of people are attracted to this bar. That's the idea. Now, that's also the thing with membrane proteins. They do this, and they are very important functionally because a lot of trafficking from one cell to another goes via membrane proteins, and certainly transmembrane proteins, that are proteins that penetrate through a membrane. Either the one side of the wall and at the other side of the wall go through the wall. Here you see one example. You know, there's a big bulb here, and this is the cell membrane, and here you see that these proteins really go through. Okay? Very often they might bind stuff, very often they transport stuff through the membrane. Okay? There's a different functionality. So these are medically very, very important. Roughly 1/3 of the proteins are membrane protein. That's the estimate. You know, of how many we have structural information? Not even 2%. Because it turns out that getting structural information of this class of proteins is a lot more difficult than of soluble proteins, the other 70% of the proteins. So, there is an information gap here. We need to find, and these are the medically very important ones, and oh yeah, we can't get these structures. So people are working very hard with other microscopy techniques and electron microscopy and all kind of things to get a handle on these structures, get them out, solve them. Still difficult. And another thing is, protein. Tell me, you should know this by now. What is normally to be expected outside of a protein floating in water? Is that hydrophobic or hydrophilic? Hydro is water, the outside crust. What you love the stuff, right? You're hydrophilic, the outside. You're hydrophobic at the inside. Now, tell you what, a membrane is not a watery environment. A membrane is a very fatty, hydrophobic environment. What do normal proteins, if they end up in a membrane, what would they feel? Like, oh bloody hell, I'm very afraid now because there's a hydrophilic amino acid, I'm in a fatty mess. That's not good. Right? So basically, all the rules of protein folding are different in the membrane. And all the technology we developed, and Tayoff matrices and Blossom, were all developed for soluble proteins. So how can we align membrane proteins? And that goes really wrong. We're not very good in aligning proteins that have different physical restraints. So we decided to build some easy technology for it. And since we had developed the secondary structure trick, we decided, hey, let's squeeze it once again, let's do the same trick, but now another way. So here we go, I show you that picture quickly, then it's done basically. So, I should have told you from that picture, you remember, right? We're going all the way back. So there is part of that, those proteins that are in the membrane, right? The transmembrane, you have a blob in water, you have a blob in the membrane, and then a blob in water. So in the membrane, these proteins should be pretty fatty, pretty hydrophobic, right? And so these are hydrophobic stretches. So it's very, and aligning these fragments in the membrane is very difficult. It goes wrong. So here is a protein sequence, and this signifies this is the bit that is in the membrane, the transmembrane bit. T is that bit of the sequence that is in the membrane, folds up in the membrane, and this is sticking out and the one side, and this is sticking out the other side. Okay, so the task here is to have these input sequences, 4 sequences. You would like to be able to really align those transmembrane fragments well onto one, right? And it turns out not to work very well if you use a standard program that knows nothing about it. So we decided, in the meantime, programs to predict transmembrane regions have become pretty good, at 90%, better than secondary structure. So we said, if we can predict it then well, we maybe can use that information and make a program that aligns these fragments better, right? So transmembrane fragments and the other fragments at the same time. Basically, we use the same thing as a moment ago. There was the slide for the 3 types secondary structure, H, E, or C. And here you see the same thing, but now just for 2 types of letters, transmembrane or not transmembrane. And people have worked out special matrices. It's called FAT, you see, for a reason, right? It sounds like fat. And like fatty and so on. It's an deviation. So we use a specialized matrix to align the transmembrane bits in the exact same way. So again, the assumption is that the patterns of exchange are different in soluble fragments than they are in fatty fragments, in transmembrane fragments, and we utilize that fact. And this really works. I'll show you one slide about the performance. Here it is, over a wide range of gap penalties, because we were afraid that, we'll be very sensitive on how we set the gap penalties here. This is when we have the combination. So each sequence has transmembrane fragments and non-transmembrane fragments. TTT, one not, and then TTT again. Here is if we use the combined way, the picture I just showed you. Here is if we don't use anything, just one Blossom matrix. And this one I think is this one. This one if we use this specialized matrix for transmembrane for the whole protein. Yeah? So here is what if we use the specialized matrix for the predicted transmembrane fragments, and the other matrix, we use 2 matrices. We use Blossom for the non-transmembrane fragments, and FAT, the specialized matrix for the transmembrane fragments. And this is if we use nothing, and this is if we use the transmembrane matrix for all letters. Both are incorrect, of course, we know it, but you see the difference. And it's stable over a wide range of parameters, right? This is always the nicer type of result, right? You see, it works in all cases. Normally, of course, this is good here, and then it goes down and under, and you need to explain to reviewers why that is to be expected. Very good, and a feature rather than a bug, and so on. Okay. So, these are just some tricks. There's one trick that will now, the last one, and then we go into what does this all mean. I'll speed up a little bit. Let me tell you one nice final story. Iteration, what was that again? You use an alignment program. You run a multiple alignment. You look at the multiple alignment, you think, maybe seeing this, I could do things differently. And you do the whole thing again with what you learned. What was a feature or a problem or a challenge in the progressive protocol? Once again, always again, by iteration, you can alleviate that problem. We're going to think again. So, iterative alignment. There are three ways you can do iteration. This is what I just said. Here. Now each dot is saying, in this case, you run a multiple alignment. This is one, and you run another alignment, and you run a third alignment, and so on. If you use iteration, if for this alignment you use information from the earlier alignment, what does this mean? This is what you hope for. If you run side last, what is the best way? What would you like to see if you run? How many iterations do you run? When do you stop? When do you feel the most at ease? When you stop running iterations, running side last? Nothing changes anymore. Convergence, that's this situation. You stop, right? Does it make sense to go on because you'll end? You will always have this alignment. So that's good. You stop. Can this happen? This is called limit cycle. This is the theory. So that means we have alignment, what is it? 2, 3, 4, 5, 6, 7 here. And you have 8, 9, 10, 11, 12, 13, 14, 15 is the same as, what did I say, 7. So if your program doesn't check it, you will spend a lot of time in your computer. But basically it will circle around through all of these alignments in limit cycle. What happens most of the time? This. How do you check for this, whether this happens? What do you need to do here, computationally? Well, you just check the previous alignment. If it's the same, you say, okay, done. If it's the same as the previous one, what do you need to check here? You go back in history and check, the before last, the before last. Then you know, right? Then you know the length of your cycle as well. Here you check, but you never find anything in history that is the same as where you are. When do you stop? When you're tired, money is up, and the computer is overheated, something like that. Huh? Or you just put, most programs have just an upper, 20 is the maximum number of iterations. If nothing converts at that moment, you just stop. Computer scientists talk about the halting problem. Can you imagine what that means? Because suppose you stop at 20, but it would have converged at 22. That's, but you're never going to find out because you stop. That's so that's a bit of iteration. I showed you already. We even discussed this quickly. This Pauline the first program, had already this iteration, updating the similarity matrix, using information from the final multiple alignment, and then do the whole thing again. I gloss over this a little bit. It's not so fundamentally important how it exactly happens. What we do here is we do consistency iteration. How can I quickly explain that without driving you nuts? You know what? I tell you, you'll be nuts in the next slide. I'll tell you more. Okay, I'll try it quickly, but it doesn't matter. Now, this is pre-processing again. Remember this slide with these master-slave alignments, 5 pieces? Now, my question to you is here, where is sequence 1 in this block? You can tell me, right? It's the top one. It's the key sequence in block 1. Is there a sequence 1 in block 2? Yeah, I see it. It's here. It's written under sequence 2. Is there a sequence 1 in block 3? Yes, it's here. So in each of the blocks, in this case, there is a sequence 1. Suppose I take the final multiple alignment. Behind the screen of each sequence, there is a sequence 1. And here I check over these 5 pre-profiles the position in the final multiple alignment. What would you expect? You would hope for the fact that in each of those sequences, alanine 131 of sequence 1 would be aligned. If that is not the case, then the pairwise alignments are not in agreement with the final multiple alignment. Already the last time we discussed that your multiple alignment will be different. Each pairwise alignment in the multiple alignment is likely to be different from the corresponding pairwise alignment. So you look at that sort of consistency, and if it's not good, then you find different consistency. It doesn't matter. We use this to say we have complicated regions that are likely to not be so correct, not so consistent. We reconsider those. So doesn't matter. Basically, we use this as a score for how consistent is each of the positions, and then we look into again if consistency is low. So here, for example, we do look in this region of the matrix as a low consistency score. Okay, so what is iffy? You do it basically. Now, what you also can do is here you see an example of iteration using secondary structure prediction. Another one. Here you see this is the proper sequences. This is the where is the proper one? One of them. Yeah, this one is the proper structure. This is what is observed, and here is what Praline is doing if you go through various iterations. And what do you see? This is stable through iterations, it's all the time there. It's just 1 to long, you see that here? But it's stable. Here it's stable, but look at this one here. In one iteration it sees it a little bit, it should be a short strand, it's too long, and then it's there and it disappears. This is what iteration can do. It changes things, and then it's there again. So basically, what should you do to use it? Here you see it, and, this may be interesting, there is nothing, it first thinks it's a helix, and later on it thinks it's a strand here, and it's not too bad, but the strand should be there. Okay. This is what prediction is to be, right? Sometimes pretty good, sometimes it's lousy. Anyway, there is another iterative program that's called Muscle. Muscle is using scissors. There's even a scissor in this picture somewhere, I think. Yeah, here. See it? Where it chops the tree and looks into parts of the sequence of it. I'll gloss over this a little bit because the point is really that there are various ways to implement iterative schemes. Reconsidering your alignment, doing it again, hopefully in a better way. Most of these methods do not guarantee that the later alignment will be better biologically than the early one. But okay, now the most important question is, what does it all mean? How good are alignments? How can we tell that one alignment is better than another? Some biologist who worked for 20 years on an alignment might have an opinion on that, but generally it's possible. What do we use to score an alignment? What does your dynamic programming do? How does it distinguish between quality of alignment? Why is that one alignment put out by the program and another one not? Because it has the highest score based on the values in the blossom and the gap penalties you set, right? Gap guaranteed, the highest score in alignment as a result. So that's great. The mathematics work, and if the mathematics will be biologically correct, then you should have your best biological alignment. But that, of course, is not the case. So this way of scoring is called sum of pairs score. Because if you evaluate an alignment, you take all these matches, mismatches, and you subtract gap penalties, and that's your score, right? That's how you do it. For a multiple alignment, it's the same thing. You can use a sum of pair scores over all pairs of sequences. The multiple alignment is just sequence 1 against 2 with a score, sequence 1 against 3, sequence 1 against 4, all pairs. You score them, add them up. That's a sum of pairs score. It's an extended pairwise alignment score for multiple alignment. And that would mean, what is your best multiple alignment that scores the highest using this score. Most programs use that as the objective function to evaluate the quality of the alignment. So a good multiple alignment program will put out multiple alignments that score high using this score. Okay? I'm telling you this is a single number for a multiple alignment saying something about the alleged quality of that alignment. Now, when we start asking people, when is alignment very good? They say, luckily there are quite a few databases. Onstrut is one, Bali base is one that is very often used, and we use it here. So we have so-called, biologists have built so-called reference alignments. They just are in the know, they claim about a given protein family, and they've made an alignment, looked at it, did correct it by hand, ran programs, another program, started looking. Some experiments were done, they knew which amino acids were functional, so they should be aligned, so did all kind of things. So then we say, this is the truth. Is that 100% correct? Let's say this. So we have whole sets, and there are in the meantime hundreds, if not thousands, of so-called reference alignments. So what do you do if you want to test your program? You take these sequences, you take them as an input, you generate for each of those families your alignment using your program, and then you start comparing your alignment with the reference alignment. And if they're all identical, then you've got a pretty good alignment. But in practice, of course, that will never happen. So there are two ways to score. Here is the very stringent, the stern scoring. What do you say? Okay. You have here a number of columns, right? 1, second alignment position, third, and so on. So you have all these columns. Now you say for each column, when is this alignment perfect? Of this column is perfectly reproduced in the query alignment. You call this query alignment, you call this reference alignment, okay? This is the alignment made by the program, and this is the reference set by biologists. So if all of those columns are identical, you have 100% hit. That's called the column score. Okay? Now, little experiment. Suppose this is a fantastic alignment. It's totally the same as this one. Only the last sequence is shifted to the right over all positions by 1 position. What will the column score do? What will the score be?
0.
Thank you, 0. All columns are a bit different. So this is all or nothing score. So some people say, well, that's harsh. That's too much. That's too critical. Let's do something else. You know what? We say we don't look at columns. We now say, okay, in this reference alignment, there are quite a number of pairs aligned in this sequence. There is this pair matched, and this pair is matched between those sequences. Maybe we have 5,000, 10,000 of such pairs in the alignment. We look at each of the pairs and just count in the query alignment how many of those pairs are reproduced. We do the experiment again. One of the sequences is perfect alignment. The bottom sequence is shifted by 1. What's the score now? 0? No, depending on the alignment, because all this one will still be correct, right? Only the pairs that include this last sequence will be off. I see difficult looking faces. So this sequence is shifted to the right, all positions. What does that mean? That all these pairs will be incorrect. Pairs that involve this sequence, right? But all the other pairs here are still okay. Depending on the size, the number of your sequences, your score will be 90%, 95%. You know? So it's not going to 0. That's the point I'm making. So these are 2 scores: column score, sum of pair score, to evaluate alignments if you have a reference alignment. Okay? And why is this annoying? Because this is the same word. This is a score that happens between 2 alignments, a reference and a query alignment. And the slide I showed you just before was the single alignment, so it's called the same thing for something different. That's annoying, right? Sum of pairs is the single score for it. All right, now. Okay, here comes the story. I'll tell you something, I call that the Charlie Chaplin problem. You have a question?
Yes, wie kent how de column score is.
I'll do this after, because I think the others have it. We'll do this after the lecture, okay? Because there are not so many minutes anymore. The Charlie Chaplin. You know Charlie Chaplin? Do you still remember him or seen it? Okay. Dressed in black. What distinguishes Charlie Chaplin? The funny walk, right? So, Charlie Chaplin was already so famous in the 1930s that people started, certainly in California, they started organizing so-called Charlie Chaplin contests. What happens? A lot of contestants come in, they dress like Charlie Chaplin, having the head and the stick. They do the funny walk up on stage, and there is a jury, 10 people giving points, and at the end of the day, the best Charlie is being selected based upon what? About what the jury thinks is the similarity with the real Charlie, right? Charlie Chaplin was no, he was known, but nobody, so he joined one of those contests in disguise. And as Charlie did his funny walk up on stage, who should get first prize? Charlie Chaplin himself became third. What does that tell you? What about, and now we are talking into multiple alignment, the jury has an objective function. They need to score using maybe this sum of pair score. They look at Charlie, do the sum of pair score of each of the Charlies, and they say, my algorithm tells me the best scoring Charlie is number 1 and the second scoring is number 2. And that's what they do. So what is happening in this jury? Apparently, their sum of pair score, their objective function is not great, if they can distinguish Charlie Chaplin and put him on top. There is something wrong with the scoring function. That's the largest problem in all of science and certain mathematics. The scoring function. What is a biological score? And this one is not the greatest. I give you an example. Here is this slide. So what did I do here? It doesn't matter alignment. This is a bunch of multiple alignments. Each dot is a multiple alignment. This is the Charlie Chaplin contest in multiple alignment. What did we do? I ran each of those multiple alignments. They're here. They're sorted according to size. How big the alignments are. Doesn't matter. This line is important. I ran each of these alignments with the most lousy option in Praline. Something we know is pretty bad. No process, nothing of the tricks, just bad. It's not too good. Then I said, how good is the alignment? You take that single, the sum of pair score, here's that sum of pair score, and if the objective function is okay, I have a box standard alignment. I calculate the sum of pair score, the score that you calculate, right? One alignment leads to the score. And I would take the corresponding reference alignment, the yellow one. If things are right, who should score higher? A bad query alignment? So, I make a bad query alignment. I score this with the sum of pair score. I score this one. Which score should be higher? Which alignment score should be higher? Of the bad alignment, you would hope, at least, that the reference alignment scores higher than the alignment you make, right? That's Charlie Chaplin contest. So what happens? Here you see your answer. This is when indeed the, how was it, value base minus praline, so what's positive? Yeah, this is when the reference alignment indeed scores higher than the query alignment, but you see how many score under the line. So very many reference alignments score lower than a pretty bad alignment that's made by a program. And what are you optimizing when you run your program? You've made it? The sum of pair score, right? So that's good. Charlie Chaplin, using this score, Charlie Chaplin doesn't become third. If 100 contestants would be there, Charlie would have become 75. Using, this is 50. Not like that. It's roughly 50. Not even third 50, right? So that's what we use to optimize our alignments. That's a scoring function. That's how good the jury is. And that's a fundamental problem, right? The Charlie Chaplin problem, I call it. And here you see a little bit how this problem evolves. The problem gets a lot. So if in larger alignments you see really that more than 50% of the alignments are third in the Charlie Chaplin score higher than the reference alignment. Not good, right? Okay. Basically it, what I wanted to tell you. I told you about different groups taking the same technology. Compare these numbers with the earlier table of problems. You'll have a space. It's the same database, the same program scored in different scientific groups. Really funny. This is the last take home. I already told it, but I'll do it now. This is, we were proud of tea coffee at the time. It was the best in the tech, not anymore. Time progresses, unfortunately, sometimes. But this was the contested PRP, was a Japanese program at the time. That was second, runner-up. So we scored the best, and then this was the runner-up. We said, okay, let's take all kinds of alignment cases, let's run the program, and let's look if the dot is here, that means that tea coffee is the better program, and if the dot is under this line, it means that PRP, the runner-up, is the better program. This is the percentage. So in easy cases, sorted again over distance, here's very distant alignment, here is easy alignment. You see that it hardly makes a difference. You see, this is that only in 53% of the cases in distant alignments, tea coffee is better. Right? And an average score is, of course, how much do we stick out. You see that when tea coffee is better, it is a bit more, the distance here is larger than the distance here. So if you score this, tea coffee comes out a lot better. This is what you have, in almost half the cases, for your situation. If this is your alignment, for this one, you should have used PRP, because that's the better program for that case. So this is the meaning of average scores and performance of always in, over 100 alignments, we score the best. This is basically what it means, right? So the average best program is certainly not always the best program in your case. So what's the take-home message? Use more methods and look at it. Yeah, don't trust anything that's too negative for the weekend. But be critical. It's good to go into the weekend.